We've spent the last several lectures looking in some detail at how developmental mechanisms give rise to the organization of complex multicellular organisms. Organisms that have many different types of cells, different types of tissues, different organs, all arranged in particular patterns relative to each other. Now, the advantage of having a more complex organization is obvious. Different cells and different tissues can specialize in an organism such that each has its own particular job to do, and each can be optimized to do that particular job particularly well. But there is a potential downside to being complex. The more parts, and the more kinds of parts an organism has, the harder it is to get them all to work together correctly. I would imagine it's a bit like the corporate world. In a small company, there are fewer employees, and each employee may have a broad range of responsibilities. It's easy for the employees to all get to know each other and know what they need to do. Coordinating the activities in the small company must be a lot easier than coordinating the activities of employees in a very large corporation where there are many more individuals, where those individuals may be much more specialized in what they do, and where getting all of those employees to do the right thing at the right time must take a lot of management. In a complex organism as well, it's necessary to have mechanisms responsible for coordinating the activities of all of the parts for the good of the whole. This idea, then, leads us to the next broad issue in biology we'll address over the course of several lectures, which is to ask how the various structures of an adult multicellular organism interact and how their activities are coordinated. In today's lecture, we're going to begin this discussion by exploring the concept of homeostasis. Now, let me begin this discussion by going back to cell function in general. All of the biochemical reactions needed for a cell to stay alive and to function well, including reactions that we've looked at for things like DNA replication or protein synthesis, other reactions we'll talk about later in the course, especially those involved in processing energy. All of these things are dependent on the particular physical conditions in which they occur. Now, the reason for this is that all of these activities are mediated by proteins. And remember that the structure and function of proteins can be affected by a number of physical and chemical conditions. Things like pH, or salt concentration, or oxygen concentration, or carbon dioxide levels. Temperature, for example, is an especially critical important factor. And this is because biochemical reactions generally run at different rates depending on the temperature at which they occur, with reactions generally running faster at higher temperatures and slower at lower temperatures. On the other hand, if the temperature gets too high, a reaction could cease to run at all because a high temperature can cause that enzyme to essentially unfold and no longer be functional. If the temperature is too low, on the other hand, actually the functioning can stop because the cell might just freeze. Physical and chemical conditions, such as temperature or pH, also can affect other aspects of how a cell functions, such as the permeability of the cell's membrane or the rate at which materials diffuse through the cell. Now, why is this an important issue for us to consider? It's important, and this may be obvious to you, because it's necessary for the cell to maintain those factors that affect its function, factors like temperature or pH, within a relatively narrow range of values, so that the biochemical reactions and other cellular processes that cell needs can work in an optimal fashion. To put this another way, we would expect natural selection to act on cells in such a way as to optimize their function, that is, to have the cell work as best as possible for whatever job it's supposed to do. But it's difficult to optimize a cell's function if conditions that affect that function fluctuate widely or occur outside of some best range. Now, this argument can be extended to the physiological function of an organism in general. The many ways that organisms make a living depend on the proper functioning of tissues, organs, and so forth, each of which may have a particular range of environmental conditions, usually a relatively limited range, within which that tissue or organ will function best. And this is where we run into a problem which really frames the subject of today's lecture. The problem is this: organisms often live in environments where the physical conditions to which they're exposed are not optimal for the functioning of their cellular or physiological processes. And furthermore, these conditions might be constantly changing. How do organisms deal with this problem? Well, some organisms simply can't. An organism that is relatively small or especially structurally simple just can't do much about it. Consider, for example, a single-celled organism like an amoeba, or maybe a very simple multicellular organism like a sponge or a hydra. Now, if you don't know what hydra are, they're actually cool, very little, very simple animals. They're freshwater invertebrates, usually just a couple of millimeters high, that are composed of only 2 cell layers. Their outer cell layer obviously is in constant contact with the environment. And so is the inner cell layer, because the inner cell layer is essentially what's lining their gut, which is continuous with the outside watery environment in which they live. Because all of the cells in a hydra, or otherwise simple and small animals like it, are in direct contact with the environment, and that environment is overwhelmingly huge as compared to the organism, we expect the physical conditions on the inside of the cells of that organism to be more or less driven by the external conditions in which it lives. And because of this, those kinds of organisms may be relatively limited in where they can live or what they can do. There's nothing they can do to change their environment, so they just have to deal with it. But larger, more complex organisms are not necessarily constrained this way. Why not? Well, because most of the cells in a complex organism are not in direct contact with the outside world. Only a relatively small number of cells or tissues, those that form the exposed surfaces, have to put up with direct contact with the environment. The majority of cells in a complex organism exist in an internal environment. This internal environment can potentially provide a buffer against the unfavorable conditions of the external environment, allowing for a broader range of options of places to live, ways to make a living in the world, and so forth. But this advantage can only be achieved if the internal environment is adequately buffered against the outside environment. In other words, if the organism can actually change its internal environment accordingly. And in fact, for this reason, much of the structure and function of complex multicellular organisms, much of what we would call physiology in biology, is specifically devoted to maintaining the internal environment of an organism within a fairly narrow range of conditions. The maintenance of a constant internal environment by an organism is referred to as homeostasis. The concept of homeostasis was developed in the 1860s, actually, by a French physician, a medical doctor named Claude Bernard, who published in 1865 a very influential book titled, and I'll give this to you in translation, An Introduction to the Study of Experimental Medicine. Actually, Bernard's book was an important contribution for many reasons, not the least of which is that it strongly made the case that medical practice should be based on experimental studies of physiology, thus helping to transform medicine into the scientific discipline it is today. It's actually remarkable that before the second half of the 19th century, medicine really didn't pay much attention to what was being learned about biology. It didn't seem relevant. Bernard argued correctly it was relevant. Now, Bernard also suggested an important point about animal physiology, and that is, he said that animals should be seen as having 2 separate environments, their external environment and an internal environment, or what he called in French, the milieu extérieur and the milieu intérieur. And to those of you who speak French well, I apologize for that. Now, Bernard argued that from a functional point of view, animals don't really live in the external environment. That is the air or the water that they would be found in. But instead they exist, and I'm paraphrasing his book in translation here, they exist in the interior environment formed by the organic liquid which surrounds and bathes all tissue elements. Bernard further argued that, and paraphrasing again, he argued, the primary condition for freedom and independence of existence, that is the primary condition necessary for survival, we'd say today, is the so-called stability of the internal environment. And that, and again I'm quoting here, all the vital mechanisms, varied as they are, that is all the way organisms work, have only one object, that of preserving constant the conditions of life in the internal environment. Indeed, as Bernard suggested, maintaining homeostasis is a ubiquitous feature of the way organisms work. Common examples of homeostasis in human physiology that you may be familiar with include maintaining a constant body temperature, something we're going to talk about a little later in the lecture, maintaining oxygen and carbon dioxide levels in blood fluids, maintaining constant blood pressure, maintaining blood sugar levels, and so forth. Now, I think it's interesting that Bernard published his book at about the same time that Darwin first published his Origin of the Species, just a little beforehand. Now, I actually think that Bernard probably read Darwin, given the sensational reception of the Origin of the Species, but I don't know if Darwin ever read Bernard. However, if more was known about physiology in Darwin's time, especially physiological mechanisms, Darwin could easily have made an equally strong case for evolution by using examples of how homeostasis is maintained in different kinds of animals. He would have been able to illustrate adaptation this way because, in fact, organisms have evolved countless different adaptations to regulate their internal environment in the face of the variable external environments in which they live. Indeed, comparative physiology textbooks today are filled with such examples. Now, instead of looking at specific examples of homeostasis in detail, I first want to consider the general principles behind homeostasis. That is, let's begin by asking what it is that an organism must be able to do in general to regulate some aspect of its internal environment. Well, first, of course, what an organism needs is to have some way to modify or control whatever parameter it's going to try to hold constant. This simply means that the organism has to have a mechanism for changing the state of that parameter. By the way, I'm using the word parameter here simply to refer to whatever it is that's being regulated, such as temperature or pH, the value of that particular aspect of the internal environment. But the ability of an organism to change some parameter is not all it needs to homeostatically regulate that parameter at a particular value. Consider driving a car. If you're driving a car, you can modify the speed of that car in a couple of ways. You can press on the accelerator, and that makes the car go faster, or you can step on the brake, and that makes it go slower. But that's not how you regulate the speed. Regulating the speed means keeping the speed at a constant speed, say 65 miles an hour if you're driving on the highway. Now, you do this not only using the accelerator and the brake to make the car go faster and slower, but you also need to have information about how fast you're currently going. You need to compare that information about how fast you're currently going to whatever speed you want to be going at, and then, in light of the difference, make the appropriate adjustment with the brake or the accelerator. So in addition to a mechanism for modifying the internal environment, homeostasis also requires mechanisms for obtaining information about the internal environment and for using this information to determine how much control to exert. We call any mechanism that modifies some parameter of the internal environment an effector. The accelerator and brake are the effectors in our driving example. We can call any mechanism that obtains information about the present state of the internal environment a receptor. In a car, the receptor is a mechanical mechanism that senses how fast the wheels are turning and converts this information to a value displayed on your speedometer. An organism finally also needs some mechanism that compares the current value of the parameter to the desired value of the parameter, calculates the difference, and then directs an effector to change the value of the parameter in the appropriate direction. Now, we can use several terms equivalently to refer to this kind of mechanism. We might call it a controller, or we might call it a regulatory system, or perhaps best, we might call it a comparator. The comparator in our driving example might actually be the brain of the person who's driving, which looks at the speedometer, knows that it should be at 65 miles an hour and makes the adjustment. Or it could be a cruise control computer, which also takes information from the speedometer and automatically will adjust at least the accelerator. So effectors, receptors, and comparators are the three kinds of mechanisms we need to have in general for homeostatic regulation. Now, there are two other terms I need to introduce at this point for us to understand how homeostatic regulation works. The first of these is the idea of a set point. The set point is the desired value, or perhaps an acceptable range of values, at which the parameter being regulated is supposed to be maintained. When driving, for example, 65 miles per hour might be the set point for the speed if you're on a highway. The second term is feedback. Now, feedback refers to the information coming from the receptor about the current state of the parameter being controlled. In our car, feedback is the information about the present speed, and we get this from the speedometer. Now, a point to note here is that whereas receptors, effectors, and comparators are all kinds of mechanisms, right? So receptors are the speedometer mechanism, effectors are the brakes or the accelerator, and the comparator is your cruise control. The terms set point and feedback refer explicitly to types of information. They're not mechanisms, they're a kind of information. Okay, well, with these general ideas in mind, let's take a look at how homeostatic regulation works. And let's take a look at another non-biological example for the sake of argument, and that is how the temperature of a room is maintained. Now, if we're thinking about maintaining the temperature of a room, first we have to ask, what's the comparator in this example? The comparator is the thermostat that's in the room. The thermostat has a set point. In an old-fashioned thermostat, that set point might be some little dial that you actually twist to specify the particular temperature that you want to be, the room to be at. The thermostat receives its information from a receptor. That receptor is a thermometer that detects air temperature and sends this feedback information to the thermostat. The thermostat is also connected to the heating system of the room, which is the effector. Now, what happens if the temperature drops below, say, the set point of 68 degrees Fahrenheit that we might want the room to be at? The thermostat detects this difference, the difference between the feedback it's getting from its receptor and the set point that it has set, and it causes the heater to switch on if, in fact, the measured temperature is lower than the desired set point temperature. The effect of turning the heater in the room on, of course, is to increase the temperature of the room. When the temperature rises back to the desired set point, let's say 68 degrees, then the thermostat will switch the heater off. The heater is no longer needed. So in this way, we've homeostatically regulated the temperature of the room. Now, we could add an air conditioner to this example as another effector and have that thermostat also turn on the air conditioner to lower the temperature of the room if, in fact, the measured value, the feedback coming from the thermometer, was higher than the desired set point. Now, this example might seem obvious to you, and in fact it is, but it illustrates an essential point, which is that homeostatic regulation depends not only just on feedback, but in fact works through what we call negative feedback. Negative feedback refers to the case when a change in the parameter being monitored, temperature in this case, triggers a response that counteracts the initial fluctuation. This is called negative feedback because a change in the regulated parameter triggers the control mechanism to prevent further change in the same direction, moving the variable back towards the set point. Homeostatically controlled systems invariably involve negative feedback. And this makes sense because negative feedback is inherently stabilizing. Small deviations in the parameter being monitored are prevented from becoming even larger deviations. Now, to clarify the idea behind negative feedback, let me contrast it with control systems that involve positive feedback. In a positive feedback system, an initial change in the parameter being measured will lead to an even greater change in the same direction for that parameter. This would be the case, for example, if we had a strange thermostat in our room that actually turned the heater on when the temperature rose above the set point of 68 degrees. Now, positive feedback systems are obviously not stabilizing because the initial perturbation is magnified. In other words, if we had our room wired up with a thermostat in this way, if the temperature rose above 68, it would keep rising higher and higher and higher. Thus, it seems that positive feedback systems would be less useful in biological control systems, certainly in homeostatically regulated systems. But actually, positive feedback does play a role in the control of some biological systems. Let me give you actually a very familiar example, which comes from childbirth in humans. When birth begins, the uterus begins to contract, and this contraction serves to move the baby down the birth canal. Pressure from the baby on the opening of the birth canal actually pushes on pressure sensors that are located here in the opening of at the opening of the uterus. feedback from these biological pressure sensors actually go back and induce even stronger uterine contractions, which it further increases the pressure, which causes an even greater feedback and even stronger contractions. This positive feedback system thus continues to amplify the uterine contractions and thus helps to bring the process of childbirth to completion. So positive feedback systems are largely involved in systems that are controlling the completion of some sort of physiological or other biological process. Well, now let's look at a biological example of homeostasis and negative feedback, and this will be involving the control of body temperature in animals. As I said earlier, temperature has a profound effect on biochemical and physiological performance, so it's not surprising that organisms have evolved a number of mechanisms for changing their body temperature and thus being able to maintain it within an acceptable range. These mechanisms are generally referred to as mechanisms for thermoregulation. Now, broadly speaking, different kinds of organisms approach the problem of thermoregulation in one of two ways. These two ways have to do essentially with the kinds of effectors they have available to modify body temperature. Ectothermic organisms change the temperature of their body mainly by absorbing heat from their surroundings. Most invertebrate animals, as well as many vertebrates like fish, reptiles, and amphibians, are ectotherms. Now, endothermic organisms, endothermic meaning getting heat from within, endothermic organisms derive most or all of their body heat from heat generated by their own metabolism. Now, by metabolism in this case, I mean heat generated by the cellular processes that break down sugars and other biomolecules in the production of cellular energy, a process we're going to talk about in some detail in the third part of the course. Mammals, such as ourselves, birds are the most notable endotherms, and there are actually some fish and even some insects that are also endothermic. Some would argue that dinosaurs were endothermic when they existed. Now, let's look just briefly at the mechanisms that organisms can use to modify temperature by comparing the control of body temperature in two organisms, one an ectotherm and one an endotherm of about the same body size. And we'll use as our example a lizard, which is ectothermic, and a mouse, which is endothermic. Imagine we put both of these organisms into separate sealed chambers, and we can control the temperature in these chambers, what I'll call the ambient temperature. Now, let's imagine also that when the organisms are in these chambers, we're able to measure their body temperature and also measure their metabolic rate, that is, how fast they're burning up sugars and so forth. As we vary temperature in this chamber from very cold to very hot, the internal body temperatures that we would measure from the mouse and the lizard would be very different. The mouse, because it's endothermic, is able to maintain a constant body temperature, about 37 degrees centigrade, but the lizard would have a body temperature that more or less is the same as the environment, more or less the same as the chamber. If we looked at metabolic rates, we'd see that the lizard's metabolism, like its internal temperature, also changes directly with the ambient temperature. It has very low metabolism at low temperatures and high metabolism at high temperatures. This is because the lizard's metabolic rate is tied to its body temperature. The metabolic rate of the mouse would also change with temperature, but in a very different way. At lower temperatures, the mouse's metabolic rate would actually be varying inversely with temperature, so that the lower the temperature, the higher the mouse's metabolic rate. The reason for this is that the mouse is regulating its metabolic rate, increasing metabolism when it's cold, specifically to generate more metabolic heat, and thus maintain its body temperature. We'd say that the mouse is using a metabolic mechanism to generate heat. Actually, the mouse has its lowest metabolism when it's at its preferred body temperature, when the outside is at its preferred body temperature, because it doesn't have to work to change its body temperature. If the temperature gets too high, actually the mouse's metabolism starts to go up again, because now the mouse has to use energy, to lose temperature, for example, through things like panting. Does this mean that the lizard can't control its body temperature at all? Well, no, actually, if we let the lizard go outside into its normal environment and measured its body temperature, we would see that it would be able to control its body temperature somewhat, not maybe as well as the mouse. What the lizard is doing is it's actually changing its position, getting into the sun if it's too cold and getting out of the sun if it's too hot. or sitting on a hot rock to gain heat from the rock, or something like that. We would say that the lizard is using behavioral mechanisms to modify its body temperature. So endotherms use metabolic mechanisms to control body temperature. Ectotherms, at best, can use behavioral mechanisms to modify body temperature. Both endotherms and ectothermic organisms actually have evolved a number of other kinds of physiological mechanisms that help them change their temperature. So, for example, in many organisms, they can change the constriction or dilation, the width of blood vessels near the surface of the skin. And the more open these blood vessels are, the more the blood can radiate heat to the outside. So when the organism is too cold, it constricts blood vessels. When it's too warm, it expands them, dilates them, and thus can modify its body temperature that way. And there are all sorts of other mechanisms, physiological mechanisms, that organisms can use. Well, now that we've examined, in general, what organisms can use to change temperatures, let's just take a quick look at how body temperature is homeostatically regulated. And in this, I'm going to focus specifically on mammals, such as ourselves, or the mouse we talked about before, partly because this basic mechanism is fairly well understood in this case. The effectors that are used in thermoregulation are the various mechanisms for changing body temperature I just discussed, especially the metabolic generation of heat or other physiological mechanisms such as changing the constriction of blood vessels, panting or sweating, which will cause evaporative heat loss, and so forth. The comparator, that is the mechanism in mammals that compares current body temperature to the desired set point, is actually a group of cells located at the base of the brain in a structure called the hypothalamus. Now, if you slide your tongue back as far as possible to the back of the roof of your mouth, and touch back as far as you can, your tongue would actually be just a few centimeters below where your hypothalamus is located. You can demonstrate that the hypothalamus is a primary regulatory center for controlling thermoregulation in mammals by, for example, experimentally removing the hypothalamus in something like a mouse and show that it's no longer able to regulate body temperature. Finally, what are the receptors? Well, actually, in mammals, cells in the hypothalamus itself are where primary information about body temperature is obtained. So the hypothalamus is serving both as a regulator, as a comparator, and essentially as a thermometer. And you can demonstrate this as well by actually implanting a small bead that you could heat up or cool down within the hypothalamus of something like a mouse. And when you heat this area up, the mouse will respond as though it's overheated, even though it isn't. And if you cool it down, the mouse responds in the other way. Well, with these pieces in place, how a mammal can regulate its temperature should be fairly clear by now. If the temperature sensed by the hypothalamus falls below some set point, say 98.6 degrees Fahrenheit in the case of humans, then the hypothalamus will trigger a number of physiological responses that are effectors that will increase the temperature. If, on the other hand, the temperature rises above this set point, then the hypothalamus will signal to other effectors that will decrease body temperature. In this way, homeostatic regulation of body temperature in animals is really pretty much the same as what we saw for homeostatic regulation of room temperature. It involves negative feedback, which counteracts fluctuation in temperature by controlling effectors that move the temperature closer to the set point. Actually, in our mammalian hypothalamus example, you can also, the hypothalamus's set point can also be changed. Various other information can either increase or decrease the set point of the hypothalamus. For example, a number of pathogens, bacterial invasion and so forth, can actually trigger cellular responses that send signals to the hypothalamus telling it to increase its set point, which causes you to have a fever. And the idea behind a fever is that it's thought to be adaptive because the raised temperature helps your body fight off whatever the infection is. Similar kinds of homeostatic mechanisms for controlling and stabilizing all sorts of parameters in an organism's internal environment can be described for all sorts of different aspects of physiology. But what I want to end with is a final point about homeostasis that we haven't yet addressed. This is the fact that homeostatic mechanisms only work if information can be transferred between the various pieces of the mechanism, from the receptors to the comparators, from the comparators to the effectors. What exactly is the nature of this physiological information? That's the question that we're going to begin to discuss in the next lecture.